Good morning, everyone. Thank you for watching this presentation. My name is Elisa Lambert and I'm a PhD student at Limoges University in France. During this presentation, I will talk about the development of a new and innovative microfluidic device for characterization and single-cell discrimination based on high-frequency dielectrophoresis. After a short introduction presenting the objectives and the motivation of this work, we'll see how high-frequency dielectrophoresis can be a very promising approach to be implemented for diagnosis purposes by probing internal cell content and operating a discrimination on cell phenotype. After showing the results of single-cell analysis, we'll see how to exploit these results in order to develop and tune a sorting system. In this project, we focus specifically on the cancer disease glioblastoma. It represents about 20% of brain tumor and it is one of the most aggressive solid tumors. It presents a high recurrence relying on a strong resistance to existing treatments. It can be explained by the high heterogeneity of the cells present in these brain tumors. Unfortunately, patients suffering from glioblastoma still currently own a very low chance of healing. The most common life expectancy is 12 to 15 months following a diagnosis. This very low survival rate is mainly due to a frequent relapse, despite the disappearance of the tumor after chemotherapy. As a consequence, this pathology is very difficult to handle. This is why the potential role of some hidden tumor-initiating cells is studied. And among these tumor-initiating cells, we found the cancer stem cells. They have quiescent property, which means that these cells are sleeping without dividing, and they can escape conventional treatment. The self-renewal capability allowed them to duplicate into another cancer stem cell. But they can also differentiate into a cancerous cell and then initiate a tumor renew. So it is a challenge to track such specific and rare cells. And how it is currently done? Biologists use tools such as fluorescent microscopy, flow cytometer, quantitative PCR, and protein array analysis to do so. However, they are faced to some difficulties. Not only cancer stem cells are very rare cells, but there are no specific immunostaining markers to identify cancer stem cells without any doubt. Current tools use a label to mark cells in order to identify them, such as fluorescent markers or magnetic markers, but they have many drawbacks. Indeed, it requires expensive equipment and the purchase of the immunostaining markers. Labels can influence cell behavior and limit further analysis. Moreover, as I said before, the controversy over specific markers for cancer stem cells drives the development of alternative cell analysis methods without markers, called label-free. These methods rely on an external force, such as optical, acoustic, or electrical force. And coupled with microfluidics, we can easily reduce the cost and analysis time with parallelization. There is no mechanical contact to prevent cells from damages, and a small sample volume is needed for analysis. The lab on chip that I present to you today is based on the dielectrophoresis phenomenon. So let me present you this physical phenomenon. A cell is a dielectric particle, which means that it does not contain electric charges that can move microscopically, but it can be polarized by an applied field. The electric field induces a dipole moment due to the redistribution of the charges at the interface between the particle and the suspension medium. In a uniform field, the polarization phenomenon redistributes the charges equally due to the uniformity of the electric field. The two opposite forces have the same magnitude, and there is no net force. When the electric field is not uniform, the field gradient introduces an imbalance, and a net force is observed, which induces the particle motion. So this is the equation of the depth force. As we can see, it depends on the electric field gradient. But but also it depends on the volume of the particle and the real part of the Clausius-Mossotti factor, which I will explain. The Clausius-Mossotti factor describes the polarization state of the particle in a suspension medium. As we can see, it relies on the dielectric parameter of the particle and the medium. It is a complex number because it takes into account the conduction and the dielectric energy losses of the particle and the medium. It also depends on the frequency of the signal generated by the electric field. If the real part of the factor is positive, then the depth force is attractive and the particles are pulled towards the electrodes as shown in this figure, and it is called the positive depth. Otherwise, if the real part is negative, the depth force is repulsive and the particles are pushed away from the electrodes, and it's called the negative depth. So, how about applying electromagnetic field to identify cancer stem cells? As a function of the frequency, the electromagnetic field allows to sense some physical property of the biological cell. At low frequency, this is the cell shape, its morphology, its size, and even the plasma membrane properties on which the electromagnetic field is sensitive. Whereas at high frequency, the electromagnetic wave can penetrate inside the cell and probe the intracellular content. As a cell is a complex biological object, for the rest of the presentation, we consider a two-shell model with the membrane and the intracellular content only. Let me show you the modelization of the real part of the Clausius-Mossotti factor thanks to the MyDepth software. We can see that there is an alternation between a negative depth and positive depth, and two crossover frequencies appear when the real part of the factor is equal to 0. It means that at this specific frequency, the cell is not subjected to the depth force. At low frequency, it is called FX01, and at high frequency, it is called FX02. So on the left side, I've done the COMSOL simulation of the cell with here the first shell, its membrane, and the second shell, its intracellular content. It is placed in the suspension medium with an applied electrical field, and the colors correspond to its intensity. The dielectric properties of this cell correspond to this curve of the simulation. The stars you see indicate the frequency of the signal used in the COMSOL simulation. So at low frequency, we can see that no field can reach the cell content, and the plasma membrane acts as an insulator. It proves that indeed FX01 depends on the plasma membrane properties, and the more we increase the frequency, the more the electric field starts to probe the cellular content, and FX02 depends on the intracellular characteristics. Cells can be individually electromanipulated according to their own dielectric properties. If we have two different cells, then they have different dielectric properties. In the same suspension medium, they will present different spectral signatures and two distinct crossover frequencies. So our aim is to identify this crossover frequency, FX02, so at high frequency, to characterize cells and use this signature as a discriminant parameter. So let me present you our Microfluidic lab on chip. We use a specific quadruple sensor for measuring the crossover frequency. And the microfluidic channel will bring the cells to the characterization area. Then the flow is cut off and the electric field is switched on. Our sensor can trap one single cell within an electrical cage. Then we can vary the frequency of the signal from a repulsive state in the center of the sensor from an attractive state. And from the motion of the cell, we can extract information about the crossover frequency. So here's a video of U87 cell characterization representing a glioblastoma cell. We start with a cell trapped in the end-depth cage. Then we decrease the frequency until observing a first motion, and finally the cell shift in the p-depth area. we can do the reverse, increasing the frequency and observing the cell repulsion. If we decrease again the frequency of the signal, we'll see the shift of the cell at the same crossover frequency. Here you can see the experimental bench in which we use the flow controllers in order to monitor the pressure applied on the cell suspension. You can see here the radio frequency signal generator, which is then amplified. The field is brought to the sensor thanks to the RF probes, and the cell suspension will cross through the microfluidic chip. In order to enrich the cell suspension of cancer stem cells, biologists submit them to stringent culture conditions. In normal medium, FBS, which is a source of nutrients, is added. Normal medium will induce a differentiation, and it is expected to have few cancer stem cells, whereas in defined medium, it will generate immaturity and select cancer stem cells. So here you can see the macroscopically effect of the culture medium. The morphology of the cells is completely different. In normal medium, cells are spread out in the petri dish, whereas in defined medium, cells form some sphere. So in collaboration with Elodie Partout, a PhD student from Culture Laboratory in Limoges, we can analyze 3 different types of glioblastoma cells. The first one is cultured in normal medium. The second one is cultured during 5 days in defined medium. And the last one is maintained during 21 days in the defined medium. So here, this is a graph of the microRNA quantification of several markers to determine the cancer stem cell present. As I said before, it is needed to cross-couple such a panel of markers. We can see that in defined medium, we have cancer stem cells, but the more the cells are in the defined medium, the more the cancer stem cell phenotype is present. So we can be sure to be in presence of cancer stem cell. We determine the crossover frequency of each different type of cells. In the graph you see 1 point is one cell signature. The experiment has been repeated several times to ensure the randomness of the analysis. So we can say that these results represent the whole cell population. So the first thing you can see is that in the normal medium, the crossover frequency of the population is spread, which reflects on the biological heterogeneity of the glioblastoma cell line. And even if we can see some overlaps in the depth signature, the difference is significative between cancer stem cells and normal phenotype. In the normal medium, the median of FXO2 is 113 MHz. After 5 days spent in the defined medium, the signature decreases to 84 MHz. And after 21 days spent in the defined medium, the signature is 66 MHz. It seems that we can follow the kinetics of the cell stemness by the decrease of the crossover frequency. In a correlation with the previous biological results of the phenotype, a lower dielectric signature can mean that the cell we are analyzing is potentially a cancer stem cell. Once we have identified the dielectric signature of the glioblastoma cell line and their cancer stem cell phenotype, we can try to isolate cancer stem cell from the whole population. So, to do so, Thomas Provence, a PhD student of Limoges University, has developed a negative depth sorting system based on repulsion. There are 2 inlets on the right and 4 outlets on the left and a sorting area. The cells are first focused by the buffer inlets, and when they reach the sorting area, different situations can happen depending on the cell dielectric properties. If the sorting frequency is near the crossover frequency, the cell will not be deflected like the blue one, and it will be collected in the first outlet. Then, the cells are deflected according to the depth force they are subjected to. And if they are strongly repelled, like the orange one, they will follow the electrodes until the fourth outlet. Then we can divide the cell population into 4 subpopulations according to their dielectric properties. Thanks to the previous cell characterization, we can choose a relevant sorting frequency to isolate the subpopulation of cancer stem cells. During this presentation, I present you a microfluidic device based on high-frequency dielectrophoresis for single-cell analysis. Coupling hydrofluidic and repulsive depth force, we are able to use a functional lab-on-chip to discriminate cell phenotype by probing their intracellular content. Dielectrophoresis is a non-invasive technique which can inform us about the potential stemness status. Then, from the cell analysis, we can select a sorting frequency to sort and isolate cells of interest. I would like to thank the European Union for founding the SUMCASTEC project with the Horizon 2020 research program and the Nouvelle-Aquitaine Council for founding the OncosomeTack project. Thank you for your attention. If you have any questions or comments.
If you have any questions or comments to make, now would be the appropriate time. If not, we will move on. Thankfully, Dr. Daniel Greiger from the University of Ulm has managed to resolve his technical issues and will hold his presentation now. So if there are no further questions, we will move on. Okay, then we'll move on. Thank you very much, Dr. Lambert.
Thank you.